Good morning, everyone, and welcome to the Intercropping Webinar. My name is Mackenzie Dune, and I'm a Crops Extension Specialist with the Ministry of Agriculture in Moose Jaw, and I'll be chairing today's event. Today, I would like to acknowledge that the presentation is being virtually delivered from Treaty 4 territory. The Ministry of Agriculture respects and has the honor of working on lands in Treaty 4 territory, a treaty that was first negotiated and entered into in 1874 in Fort Qu'Appelle, Saskatchewan. It is important to recognize that the land on which we are virtually hosting this is the traditional ancestral lands of the Cree, Ojibwa, Saulteaux, Dakota, Nakota, Lakota, and home of the Métis people. I would also like to acknowledge that we respect and honor all other treaty territories that we work on through provinces. Treaty 2, Treaty 5, 6, 8, and 10, and are committed to move forward in partnership with the Indigenous nations in the spirit of reconciliation and collaboration. CEUs are going to be offered for CCAs on this webinar. In order to get the credit, you need to attend the entire event live, and there will be a QR code at the end of the presentation to collect credits. If you happen to miss the code, just let us know by email afterwards and we can get you your credits. Certified crop science consultants wishing to collect CEUs for today's session may submit a CEU request through the CCSC program self-reporting database at ccsctracking.ca. Also, I want to mention that the 2023 Crop Diagnostic School will be held on July 25th and 26th in Indian Head. More details are coming out soon on the event, so stay tuned. Each presentation will be followed with a live Q&A and the main hub for today's webinar screen. And the main hub for today on your webinar will be the screen. And we will share relevant and downloadable handouts with you in the chat box area. The entire session will also be recorded, so if you miss anything or wish to revisit any of the presentations, you will be able to. Recording links will be sent out later today in a follow-up email. Also, an evaluation will pop up at the end of the webinar this morning. Please ensure you fill out this evaluation, as this helps us to determine what topics you want to learn more about and helps prioritize research for the future. We also have a few housekeeping notes today to keep in mind during the webinar. So if you have any questions during any of the presentations, please type them into the question box on the right-hand side of your control panel. We will try to get to as many of the questions as possible, but may need to limit them due to time. Your questions will remain anonymous and they'll only be sent to me. During the presentation, you will also be muted so that we can ensure there is no feedback that will interfere with the audio. So first up, we have Dr. Michelle Hubbard. Dr. Michelle Hubbard will be talking about disease control and agronomy practices involved in chickpea and flax intercropping. Michelle has worked as a pulse pathologist with the Agriculture and Agri-Food Canada and Swift Current since 2017. Her research focuses on ascochyta blight of chickpeas, anthracnose of lentil, and root rot in pea and lentil. She is interested in innovative, sustainable, and practical disease management. Thank you so much, Michelle, and we'll start whenever you're ready.
Okay, can everybody see and hear me? All
Yes,
right.
we can, Michelle.
Thank you. Okay, so thank you for inviting me to talk about intercropping. That's one of my favorite topics and one that I'm excited to be talking about today. So I'll be focusing on chickpea flax because that's the combination that I've done the most work on. However, I'll also, as I'm first in this webinar, introduce intercropping more generally and talk about some other crop combinations beyond chickpea flax. So first off, what is intercropping? Intercropping, most broadly, is growing two or more crops together at the same time in the same space. There's many ways that this can be done, including mixed or alternating rows, which those two I will talk about in some detail in the chickpea flax portion of the talk. And then there's also other combinations such as strips, where there's more than one row of each intercrop side by side. Relay cropping, where one crop is seeded first and grows for a while before the second crop is added. Intercrops can also be harvested for grain that's separated, or they can be grown for forage, in which case the grain is not separated, and that changes the considerations. The diagram I'm showing here is from a recent scientific paper about chickpea flax intercropping and for yield and disease management. And this is not my work, but it's exciting to see that there is a publication out on the topic, as there's certainly a need for more research. And I've just put up the information about the publication if you are interested. So why would you want to consider intercropping? The most well-known perhaps reason, or the one that often comes to mind first, is to increase yields. And this is often thought of in terms of overyielding. And overyielding occurs if there's what's known as a land equivalency ratio, or LER, that's greater than 1. And so an LER is basically a way of saying that by having 2 or more crops together in the same space, the yield per unit area is greater than, or the land that is required would be less than that would be needed to get the same yield from those same 2 crops. So to calculate this, if we go with the chickpea and flax example, this would be the yield per unit area of flax in the intercrop situation divided by the monocrop yield of flax rather, and then that would often be a fraction, like often but not always, intercropping would reduce the yield of each of the intercrops. So say that fraction might be 0.6. So you got a little more than half the flax yield out of an intercrop as you would out of a monocrop. And then for chickpea, say you got 0.8 of your yield. So for both crops, if you consider them individually, the yield went down. But if you add 0.6 and 0.8, you would get 1.4. So that's greater than 1. So you're coming out ahead. Other reasons to intercrop are many, and they could include disease management, either of foliar or below ground diseases. Then they could be reducing the need for nitrogen fertilizer, especially if one of the intercrops is a nitrogen fixing crop, increasing diversity, improving soil health, reducing lodging of one or both of the crops, perhaps weed management and risk management. If one crop were to fail, the other may do well, and risk management in terms of managing weeds or other pests or other agronomic problems such as lodging, or reducing risk in terms of reducing your input costs through reduced fertilizer. And some considerations when thinking about intercropping. So these are things that some of you listening as farmers may know far more about than I do, as I'm not a farmer, but this is just an introductory list if you're new to intercropping as to things that you should think about and consider before you start or before you expand or try a new combination or a new system. So first, when and how to seed. Do you seed both crops at the same time? Do you seed them at the same depth? Or might the larger seeded crop go deeper than the smaller seeded crop? And what equipment might you use to do this? The picture here was kindly provided by Taylor Snyder via Twitter, and he provided a lot of pictures that illustrate these considerations really nicely. So I'll be showing some more of these in the coming slides. Other considerations, some of which I'll go into in a little more detail in the next few slides, are things like the seeding rate of each crop, what fertility you'd want to use, how you'd manage weeds, timing of maturity and planning for harvest, seed separation, and storage of the crop, either separately or together. So seeding rates, often where people tend to start, or historically tended to start, is cutting the rate of each crop in half. And so using 50% of a reasonable monocrop rate for each crop. But that, of course, is not the only option. You could plan for one crop to dominate or anticipate that it may dominate and move the seeding rates up and down accordingly. Common combinations are things like 30% of a monocrop rate and 70% for the other crop. Of course, the seeding rates don't have to add up to 100% though. For example, in the chickpea flax trials that I'm involved with, we keep the chickpea seeding rate constant regardless of how much flax there is or isn't. And so this is always an option, or just reducing one rate slightly so that the rates add up to some number that's greater than 100%. Fertility is an important consideration, especially nitrogen. Many intercrop combinations involve at least one nitrogen fixing crop, and this can reduce the need for nitrogen, which can, as nitrogen costs can be high, this can have a cost benefit and also potential environmental benefits. The fertility can obviously increase or decrease the competitiveness or dominance of one crop. The picture here that was provided by Taylor Snyder, again, really nicely illustrates this dominance. So it's a three-way mix of oat, flax, and pea, where the oat really dominates. That you have to kind of search in the picture to see the tendrils of some pea in there, and then the person's hand is making the flax obvious, but the oat is what's predominating. And then harvest. The crop combinations should mature at the same time. If they don't, it can be very difficult or just not feasible to harvest one crop first and then go back for the other one, or leave one crop to wait and risk seed shatter and issues associated with that. So that is an important consideration, and the relative maturity timings of different crops can differ depending on the moisture level and the year and the location. So that's something that you need to consider in terms of planning appropriate combinations. And then combine settings to successfully harvest seeds of different sizes from different crops also need to be considered. Then once the crop is harvested, seed separation is an important consideration. Typically, good intercrops, if they're being grown for grain, of course, you need to separate the seed, and often this is done on a basis of size. And then you can use gravity or sieving to separate out the seeds. And you can also use shape. I've seen this done where seeds that are similar size can be separated based on the speed that they move through an apparatus, and so they could be separated that way. In any case, you should have a plan before you set out to do an intercrop, how you'll separate it. Will you do it yourself? Will you do it before you store the seed? If you plan to store mixed grain, how might that impact moisture and the preservation and shelf life of the grain? And so these are all things that you need to consider. And if you're new, perhaps start small so that you have some time to work these things out before you go to a really large scale and potentially have issues. Now I asked for some examples of farmers who have actually tried intercropping at a commercial scale, and a few people were kind enough to provide me some information over Twitter or email. And I wanted to include this to both because I think I, as a researcher, can learn a lot from farmers. I don't farm, and my knowledge of equipment and of the experience of being a farmer and what farmers actually need and want is limited. And so I want to increase that. And then I want to facilitate through this webinar sharing information so that you can hear a little bit from each other. And of course, can talk to each other and about experiences. So Tyler Snyder said he has tried intercropping in 2020, 21, and 22, and has tried a range of different intercrops, all of which included pea, which is lovely to see for me as a pulse pathologist. Initially, he said that his interest was something to prevent soil carbon loss after peas were harvested. And so he was looking at a cover crop, but then eventually decided to go with an pea oat intercrop instead on recommendation from Kevin Elmy. And so he tried this in 2020 and was really happy to see that the pea quality and the pea yield was improved. And in fact, the peas yielded better than a monocrop. So not only was the LER over 1, but the peas actually in an intercrop actually had a higher yield. In 2021, he expanded to pea and barley. And in 2022, he tried that three-way mix that we saw of pea, oat, and flax, where the oat dominated. Then an example from Grace and Meaden, where he tried a forage mixture of pea, so again, a pulse or a nitrogen fixer, and along with oat and triticale back in 2020. And when I asked him about why he was interested in trying this, he said that he does organic production and wanted to increase symbiotic relationships, increase yield without extra inputs, and that he was hoping that the soil health would be improved through the addition of triticale, which might increase soil fungi. He was also hoping the feed would be more palatable. And then he gave some information about the seeding rates of 1 bushel for oats, half a bushel for pea, and half a bushel for triticale, and no fertilizer was used. And there's a video of this operation of the mowing on Twitter. Maple pea and camelina is another commercial combination, and Sheldon Dowling has tried this. He found that the maturity lined up well, and because of the seed size difference, they were easy to separate. And he seeded the peas at 120 pounds per acre and the camelina at 4 pounds per acre. Another combination that is one that I'm interested in exploring in future research is lentil intercropping, and in this case, lentil and flax. So Avery Rusk of Triple Rusk Farms has tried this combination last year, and he didn't use any nitrogen fertilizer, seeded the lentils at 90 pounds and the flax at 12. And here's some more pictures of how that looked a little later in the season. He's also done 9 years worth of chickpea flax intercropping, so has obviously been happy with the results, and seeded the chickpeas at 140 and the flax at 20 pounds per acre. And this leads nicely into the small plot research that I've been involved with on chickpea flax. So the preliminary work on small plot chickpea flax was started by my colleague Lana Shaw, who's based at the Agriarm site in Southeast Research Farm in Redvers, Saskatchewan. And she and Bill May of Agriculture and Agri-Food Canada and Indian Head did preliminary trials with chickpea and flax, initially trying reducing the chickpea seeding rate when flax was included, and determined that it worked better to keep the chickpea rate high at a monocrop rate. And as a result of that promising preliminary work, some of which showed really strong disease management potential with the intercropping in years with high disease pressure in redvers, we were able to use that preliminary work and build on it and get an ADF funded research project that began in 2019 and is now nearing completion and ran in 2019, 2021, and 2022, and has 5 sites across Saskatchewan. It includes my site in Swift Current, which is a typical low-risk, dry chickpea growing area, and then Saskatoon at the University of Saskatchewan in Risk Group 2 for chickpea insurance, and then redvers and Indian head that are pushing the limit of where it would be insurable to grow chickpeas. They're both wet, and so it could be difficult for chickpea to mature within the time span that they have for the growing season. And then Melfort, which is even wetter and way outside of where you would normally grow chickpea, at least as a monocrop. And we included that, although it may not make sense to grow chickpea there, though I know that a few people have in drier years successfully intercropped chickpea and flax in that area. We wanted to include it to push the system and to see what would happen in a site that was likely to be wetter. When and how might this system fail? So that we can do silly things as researchers, so that, and then tell you as farmers or agronomists about it, so that farmers don't have to repeat mistakes or will know what may or may not work. So why chickpea flax? The biggest reason from my point of view is disease management of ascochyta blight. And I'll have quite a few slides on that as the presentation progresses. And then there's also agronomic reasons. So as you're likely all aware, chickpea is an indeterminate crop, meaning that if moisture and conditions permit, it will just keep growing, keep flowering, stay green. The seeds won't mature or won't produce seeds. And eventually in Saskatchewan, frost will come and you won't get a yield. So getting the plants to mature in a timely fashion, the thinking is that flax can dry down the soil and provide this stress and increase the chances of the chickpea maturing and producing non-green seed by the end of the season. And this could expand the area of adaptation. So while it may still not make sense to grow chickpea in Melfort or in Manitoba, it could expand the area. And that could be really helpful, for example, to get another pulse in the rotation. In some of these areas, root rot could be a huge problem for pea and lentil, but not so much for chickpea. And if intercropping allows producers to get chickpea into the rotation, that could have a big benefit of another way to include a pulse while still keeping that long rotation that's recommended for Aphanomyces root rot away from pea and lentil. And it could also be a form of risk management. So reducing your risk in those borderline areas where chickpea could be a really risky crop, and reducing the risk of input costs needed for fungicide, for example, and perhaps for nitrogen fertilizer. So chickpea, historically in Saskatchewan, the acres have been quite volatile. And this is a graph showing the seeded and harvested areas over time. So chickpeas were introduced, the acreage shot up, and then ascochyta blight set in. And prices also play into this, that the acres tend to be cyclical. And this is, of course, due to ascochyta blight risk, as well as some of the other risks I mentioned. But ascochyta blight is a huge problem for chickpea growers. It's caused by the fungus Ascochyta rabii, which is highly destructive and can completely destroy yields. It is spread both by small asexual spores, which is the picture for me on the left-hand side. Anyway, the microscope picture with two oblong structures in it. Those are asexual Ascochyta rabii spores. So the seeds of the fungus, the way it disperses itself in the growing season from plant to plant. And it can also reproduce sexually, so that increases its diversity and allows it to evolve and overcome both the partial genetic resistance in chickpea varieties and to develop insensitivity to fungicides, especially group 11 or strobilurin fungicides, which are a major way that this disease is managed. And fungicides are costly, and can fail despite 4, 5, 6 applications in a season. The disease can still be intense. And so looking at something that could be used in addition to fungicides, perhaps reducing the need for fungicides, or in combination with new varieties of chickpea, makes a lot of sense. So that's the thinking behind intercropping. And then some of these pictures here illustrate what ascochyta blight looks like and how it progresses. So typically it shows up first as teeny little black dots. So the picture, the top picture of leaves where there's very small black dots, those are just the beginning symptoms, and they are reminiscent of dirt. So if you are looking, scouting for the disease, and you brush a leaf and it comes off, obviously it's dirt. If it doesn't, though, chances are it's ascochyta blight. As time passes, those dots get larger, and that's what the picture from Lana Shaw shows, is those lesions expanding. And then there's a close-up of a lesion that has expanded quite a bit with a red circle and a red arrow showing the typical, though not always present, concentric circles. And what those concentric circles are the pycnidia, or the fruiting bodies, the structures in which spores are produced. So inside each of those tiny little black dots, there's many, many, many spores that are then dispersed by wind or rain from that leaf to neighboring leaves, stems, or pods, and or to neighboring plants. And in this way, the disease can spread field to field, but also with these asexual spores, it's typically within fields. So it can start off in patches and then expand. This slide shows as the disease progresses, it can damage stems. And this is where a real problem sets in, that as the stem gets encircled or girdled, it can break. And then all the growth above that point is lost, including any potential for pods. And so you can see that in a really nice picture here from Betty Johnson, one of the plant health officers. The disease can also spread to pods. And here's a beautiful photo from a pathology point of view of concentric circles forming on a chickpea pod taken by Lu Min Luan. And it can also be seed-borne. So if the disease is on the pods, it can then infect the seeds and potentially be passed on to the next generation of plants. And so that's something that you really want to prevent, and you also want to use clean seed, so seed with no or virtually no ascochyta blight infection. So after that introduction to ascochyta blight, the big problem we're hoping to solve, or address at least, here's some background about the small plot experimental design. So as I mentioned earlier, we use chickpea at a monocrop seeding rate, which we think of as 60 seeds per meter squared, aiming for 40 viable seeds. And that works out to a little over 200 pounds per acre. And then the variety we used is CDC Leader, an older variety now, but the most widely grown, and it's a Kabuli variety. And then there's 3 factors in this experiment. One is flax seeding rate, so 0, 75, 150, 300, or 600 flax seeds per meter squared. And then we use either mixed or alternating rows. So either growing the chickpea and flax together in the same row, or growing them side by side, so a row of chickpea and a row of flax, etc. And then we've used either no fertilizer, which actually does have a small amount of nitrogen fertilizer, but that's across all the plots, or the N60 treatments that have additional nitrogen fertilizer side banded. Here's a picture of what alternating rows looks like in Swift Current in 2022. And so this was the 150 seeds per meter squared of flax. So that's the second lowest. The rates were 0, 75, 150, 300, and 600. So this is kind of middle. And then this is mixed rows of the same flax seeding rate. And if you notice the stick with something white on it in the background, that's a hobo data logger. And I'll touch more on that a little later on. Basically, they're present to monitor the temperature and the relative humidity in the canopy. So to move to results, some historical results that I've presented before in other presentations. In Melfort in 2019, the disease was relatively low. So out of a possible scale of 0 being healthy to 9 being a dead plant, the disease was in the 1 to 3 range. And we found that flax seeding rate made a difference. If there was any flax, whether it was that lowest rate up to the highest rate, that didn't make a difference, but having any flax reduced the disease. And here you'll see that I've translated the flax seeding rates. It's still 0, 75, 150, 300, and 600, but I've translated it into pounds per acre. And then row placement did actually make a difference, and there was more disease in mixed rows as opposed to alternating rows. This is a little surprising to me because I would have thought that mixed rows would have less disease because they would perhaps provide a physical barrier to prevent spore spread from a plant to plant within a row, whereas in alternating rows, the disease has the potential to just spread up the rows. So this was an interesting and surprising finding. In Melfort last year, the year that just ended, 2022, disease was extremely low, 1 or less than 1, and there was no difference between any of the treatments. In Redvers, the site in the southeast of the province, we found in July of 2019 that flax seeding rate lowered disease and that higher flax seeding rates lowered disease more than lower seeding rates. But we found that in August, this is still the July data, but in August, there was no difference. The disease was similar across all treatments. In this past year, so 2022, there was again disease in Redvers. In early August, it was still fairly low, but there was no differences between treatments. As August progressed, differences did appear, but surprisingly, or contrary to some other results, flax seeding rate didn't make a difference. So there was no benefit here to intercropping. However, using nitrogen did increase the disease, and mixed rows decrease the disease compared to alternating rows. So that differs from those Melfort results I was showing you a couple slides ago and matches up more with what I would have expected to see. Again, later in August, we found there was no difference between flax seeding rates, but mixed rows still had less disease relative to alternate rows. And so now to get into something that to me is very interesting and that Melissa will touch on more in her talk, are exploring when this works, why does it work? And this is both scientifically interesting to us as researchers, but perhaps very useful for farmers when there is more information, because if we know why it works and how it works, that could allow you to plan your cropping system better and increase the chances that it will work. So there's a few possible mechanisms that it could work by. One that is last on this slide, but which Melissa will discuss, is perhaps the presence of flax somehow, perhaps through arbuscular mycorrhizal fungal networks, trigger chickpea defenses. Another that I touched on earlier is a barrier to spore spread. And so this just means a physical barrier. if you remember how I said Ascochyta spores can spread from plant to plant and from tissue to tissue. Having flax, which can't acquire Ascochyta blight, in between chickpea plants, as wind or rain splashes, spread is just less likely and can happen less. And this, one would think, would happen more in mixed rows than alternating rows. And also with the plant defense triggering, mixed rows may be better because they might allow more thorough intermingling of the plants and hence better defense triggering. Then another mechanism that we are actively exploring is canopy microclimate. So diseases in general like the environment to be moist. And this makes sense if you think of a disease coming from a spore, so like a plant seed, that seeds and spores need moisture to germinate, and then they're vulnerable once they germinate. So they need moisture to survive as seedlings or as fungal germlings, as little hyphal tubes, little vulnerable baby fungi that will be on the outside of the plant initially and then look for a way in. So especially when they're on the outside, having moisture in the air or moisture on the plant surface is essential for their survival. So what we did to look at this idea and test this hypothesis is we put hobo data loggers, which are these structures shown in this picture, and they're devices that measure the canopy microclimate. So every hour they would measure the temperature and the relative humidity, and we'd move them so that they were we would only put them out once the plants were big enough for it to make any sense, and then we'd move them up as the plants grew, and we'd compare treatments. So here's some of the results from this past year. And so they, as you can perhaps appreciate, measuring every hour in many treatments and many sites is a lot of data to go through. So we haven't finished the process of analyzing it all, but from some preliminary work I've done looking at relative humidity, it looks like the monocrops are actually drier. So their relative humidity in this graph was down around 80%, whereas in the high flax rates intercropping, the relative humidity was more like 82, 83%. And so is this enough to make a difference? And it's also going the wrong way. It's saying that there's humidity in a treatment where, based on some of our results, we'd expect there to be less disease. So does this mean relative humidity is not part of the picture, or does it mean perhaps, depending on the site and the conditions, sometimes it's part of the picture, but it can't be the whole picture? Also, this was in a situation where there was no difference in disease. So we really need to look at more data to get a more complete picture. When I broke down the data and looked at shorter time intervals, sometimes the results, the trend is similar, but which treatments are significantly differed. So the previous graph was the entire season, and now this one is just looking at a period in early July. And so we found still the same general trend, but this time it's the mixed rows that are moister as opposed to the alternating rows. To add to the complication and the confusion is back in 2019, we found in red vers, looking at a time period right before there were differences in July in disease, we found that the higher flax seeding rates was higher humidity. Sorry, I misspoke. That was different in that there were disease differences, but the trend was going the same direction as this previous slide, where the higher flax seeding rates had more humidity. And in the same year in 2019, I'm not showing the graph, but the opposite actually happened in Melfort. So that perhaps argues for different site-to-site differences. Maybe something different happens in the north versus the south. But as I say, we have a lot more data to wade through. Now, if you remember, red vers had no intercropping itself had no benefit in terms of disease, but it had an enormous benefit in terms of yield. So this is a really exciting result because this is not land equivalency ratio, but straight up yield and just chickpea yield. But we found the monocrop, where it's a 0 flax seeding rate, was quite low. And then at a moderate flax seeding rate, especially in the mixed rows, the yield was much higher. So as around 450, 500, up to something like 1100. So it's a huge difference. And then when mixed rows, as the flax seeding rate increased higher, the yield dropped off. In alternating rows, that didn't happen. We also found that nitrogen had a significant impact, and flax seeding rate interacts with row arrangement, as this graph shows. And so in general, nitrogen fertilizer would favor the flax. So flax would grow more relative to the chickpea and would be perhaps the equivalent of a higher flax seeding rate. Then some results from Indian head yield in this past year. And so this shows both flax yield and chickpea yield. Chickpea yield, it's pretty stagnant, but pretty high. So similar to the red vers yield at its best was across the board for seeding rates in Indian head. And then, of course, flax seeding rate, as it went higher, as one would expect, there's more flax yield. And having nitrogen in the mix increased the flax yield, as I mentioned previously, that, as one would expect, favor flax. Another sometimes benefit of intercropping chickpea and flax is a reduction in green seed. And so this benefit only shows up when there is a substantial amount of green seed to start with, as you'd expect. And so the pictures up top here show an example from red vers in 2019. And so that is the red 2019, or the green bars show the data. So you can see at 0 flax, the percent green seed is 17, 18%. And then as flax is added, those percents don't go to 0, but they drop a lot. And the picture visually illustrates this. And then you can see in some other years, like red vers 2017, similar, a big impact, but then some years not so big. So 2021 in Indian head, there's maybe a trend towards less green seed. And then if we look at the 2022 results in Indian head, otherwise this is exactly the same graph. It's just the green seed was super high this past year in Indian head. And so what happened there was it was dry early in the season, but then wet later on. So that meant that by the time the rains really came in July, the flax was at a point where it could no longer drink up that moisture. And so that meant that the chickpeas were left with a whole bunch of moisture late in the season. Hence, they didn't mature and had a lot of green seed. But because the flax was so far along, it couldn't have as strong an impact as it might have been able to have had it been wet earlier. So there is perhaps a trend towards reduction in green seed with higher flax seeding rates, but there's still a lot of green seed. And then here, just switching the flax seeding rate units. Then moving on to Melfort this past year, where there was no difference in disease, there were differences in yield. So unsurprisingly, yield is less in the higher flax seeding rates for chickpea, as there's more flax competition, and the flax yields go up. The overall yields maybe go down slightly, but the LERs are over 1. They're not dramatically over 1, but you're not losing out overall. And then if you look at the chickpea yield broken up in more detail, you see that there is that the same general trend: more flax, less chickpea, but at those higher flax seeding rates, the alternating rows perhaps compete less with the chickpea and allow there to be more chickpea yield. Then something really interesting that meshes perhaps with green seed data is maturity. So these numbers on the y-axis, the vertical axis, are Julian dates, so the day into the year. And so a smaller number means the chickpea matured sooner. And so here there's a big difference, especially with the no N. So the treatments are written across the bottom, and at first it's no N with different seeding rates of flax, and then with N. And so especially without nitrogen, the chickpea matured up to 10 days earlier, which, depending on the season, that might be the difference between having a crop and not. We also found that higher flax seeding rates led to earlier maturity, and mixed rows led to earlier maturity compared to alternating rows. And just changing the units for the flax seeding rates. So that brings me to the end of my content on chickpea flax. And now I know I don't have a huge amount of time, so I will go a little bit more quickly through these other systems of intercropping that I want to talk to you about. One is pea brassica intercropping as a strategy to manage root rot. And so this work is led by Dr. Shama Chatterton of AAFC Lethbridge. And so first off, as you all are likely well aware, root rot is a serious problem in pea and lentil, and it's caused by a range of fungi and oomycetes. These pictures show how destructive it can be, completely killing some peas in Redvers at Southeast Research Farm and causing severe root rot. The organisms that cause it are, the most important one is Aphanomyces euteiches, and then many Fusarium species, as well as Pythium and Rhizoctonia are also involved. The oospores of Aphanomyces are a huge problem. They're long-lasting resting structures that can survive in the soil for many, many years. And they mean that long rotations away from pea and lentil are really the only proven strategy. However, in theory, intercropping with a brassica could help. So the theory is that brassica species contain glucosinolates, which can then break down to isothiocyanates. And this would happen as the roots or the plants decay and break down themselves, which might be a problem because in intercropping, the brassicas are alive. However, in theory, it seemed worth testing. So we did test it over a number of years. Back in 2019, this is some of the data from Tabor, Alberta. Overall, what we found is that root rot levels were never reduced. And this might be, as I mentioned, because the brassicas are still alive. It might also be because of challenging conditions, such as hot, dry conditions in 2021, very severe root rot in 2022, or it may just not work. However, there were benefits in terms of yield in quite a few sites and years. So in this table where it shows LER, the numbers for some of the treatments, such as pea and canola 70-30, pea and canola 50-50, pea mustard 70-30, and pea mustard 50-50, the LERs were all over 1. Then in 2021, again, LERs were sometimes, depending on the combinations, over 1. And all of this was done in mixed rows where each crop was seeded at 50% of the monocrop rate. And again, and then in Lacombe, some brassicas actually did better as an intercrop than a monocrop. So not just a positive LER, but like a straight up yield benefit. But then some combinations didn't do so well. And again, this was a hot, dry year. So there's very worthwhile to do this, both to find out there is no root rot benefit in many situations, but some of the combinations perform differently in terms of the yield benefit that they did or didn't have. So that's useful information to have. And then in Swift Current in 2021, again, we had some over yielding, but no root rot benefits. Then the final crop combination I will talk about today is lentil and camelina. And so this was work done by Bill May out in Indian Head. And it was a fun day for me to go and see it. So I was able to go on a weekend and with my little daughter, who was a little over 1.5 at the time. And so the picture here shows first camelina just by itself, and then the intercrop. And so my interest in this for the future is I'm looking at lentil intercrops in general, or I hope to look at them perhaps as a tool to manage anthracnose of lentil. And so I'm really interested in seeing which combinations may or may not work well. And so there was no disease in this trial. So from that point of view, I was disappointed, but the results are hopefully of interest to you. And so this system actually seemed to work quite well. So here there was a reduction in weed biomass. So the two monocrops are the last two treatments, camelina monocrop and lentil monocrop had the highest weed biomass. And then some of the intercrops, well, one of the intercrops rather, had a statistically significant reduction in weed biomass. And the treatments that Bill used were 3 different seeding rates of lentil. So the monocrop rate of 120 seeds per meter squared, and then 90 and 60. So 3/4 and 1/2. And then camelina, he kept at a monocrop seeding rate of 5 kilograms per hectare, regardless. And then he used 2 different rates of nitrogen, so either a low or a high, 21 kilograms per hectare or 75. And he found that nitrogen in general had a bigger impact than seeding rate. And in terms of yield, there were some benefits. So to illustrate that, I've drawn this line with the lentil monocrop yield, and you can see that some of the treatments there was less lentil, yes, but some there was, despite a full crop of camelina being there was no reduction in lentil yield for some of the treatments, especially 2 of the low nitrogen treatments. And then when you look at camelina yield, it was always either the same or better when lentil was included. So that's quite exciting. And so that brings me to the end of the data that I have. So to wrap up, intercropping is in fact being done in the Canadian prairies at a commercial scale, and it has a lot of potential benefits, including foliar disease management, yield benefits, benefits in maturity, however, not benefits to root rot. Hence, it's not a cure-all. It doesn't always provide yield benefits. It doesn't always provide foliar disease benefits. And that leads to a need for more research to understand and help you as farmers better predict when it will have a benefit and when it may not. And you should use combinations that work well together. So don't just do intercropping for the fun of it, but think about what issues you're trying to solve and do crop combinations work together. And that requires planning. And I see a need for farmer to farmer, farmer to researcher, and researcher to farmer knowledge transfer so that we can collectively do a better job of incorporating intercropping in a way that makes sense and solves problems. And that, of course, means that I, as a researcher, have lots of work I'd like to do, and so do my colleagues. And with that, I'd like to acknowledge the many organizations that have funded this research and the scientific and support staff from the many sites, including Southeast Research Farm, Alberta Agriculture and Lakeland College, and the many AAFC sites. And thank you very much for your attention. And I would be happy to answer any questions if there is still time.
Awesome. Thank you so much, Michelle. That was a great presentation. So there are a couple of questions that came in over the presentation. So the first one is, does Ascochyta reproduce sexually or asexually?
That's a good question. It does both. So during the growing season, it cycles asexually and will produce those small pycnidia asexual spores that spread on a relatively short scale within fields, but then it can also produce sexual spores. And that's not the case everywhere. For example, in Australia, they haven't seen sexual recombination, but in Canada, there's both mating types and the pathogen can reproduce sexually, and that increases the chances of what's already happened once, where there's partial genetic resistance in chickpea varieties, and then the fungus can evolve so that it overcomes that resistance. It also helps with overcoming or becoming insensitive to group 11 fungicides or strobilurin fungicides, which are widely used.
Awesome. Thank you so much. The next question is, was there any height differences in lentils when intercropped?
That would be nice to know. In the in Bill May's study, I haven't seen that. I just haven't seen any data for it. I think they did. Bill, I believe, didn't collect that data. And so in future work that I'd like to do, that would be an important thing to measure. If I remember correctly, there was a trial that was done both in Outlook and in Redvers where they did measure height and didn't find a difference. But what I would expect could happen at least some of the time is that lentil could be taller in the intercrop, that as the companion crop is almost certainly going to be taller than lentil, lentil would be looking for that light and or held up by the companion crop. And so then it would be taller and perhaps the lowest pods would be farther off the ground, and that this could help with harvestability.
Awesome. Thank you. The next question is, are there any thoughts to using specific herbs instead of traditional crops as intercrops?
Using herbs. I don't have plans to do that. It could perhaps work. Yeah, no, it would be worth exploring, but I don't know much about it, but I imagine it could be done.
Awesome. Thank you. I have one question of my own here. So you talk about the effect of maturity with intercropping. What interaction do you think is happening between the crops that is affecting maturity?
For chickpea and flax, what I think is happening is that when there's an intercrop, it draws down the water more in the soil and provides a drought stress, and that triggers the chickpea to mature earlier. So chickpea will just stay immature so long as it has water to keep growing. But if it has less water due to flax being there to drink up some of that water, that could trigger earlier maturity.
Perfect. That makes a lot of sense. Thank you. All right, one last question. Looking at intercropping on irrigation fields, is there any research in this area? Are you concerned in regard to the higher yield potentials and nutrient requirements?
Yes, there's a very small amount of research on it. The last year there was a trial on lentil intercropping with camelina under irrigation at Outlook. And there's been irrigation studies actually in the US on chickpea flax, both published and unpublished. Some of that work that I mentioned at the very beginning of my talk out of the US, some of those sites and years were irrigated. And so from my point of view as the pathologist, I'd like to do it for research, not necessarily because I think would make sense as something farmers should do, but because in order for me to find out, does intercropping help manage disease, there needs to be disease. And sometimes there is no disease if it's dry. So irrigation would be a way to get disease going. And then if there's disease, you can see is there differences between an intercrop and a monocrop. And then I think one of the results that was found at Outlook last year is there was a lot of lodging, and there was also a lot of Stymphalium blight, the foliar disease in lentil, but no data as to whether it was any different between the treatments. So that would be interesting to know. And then the other part of the question was nutrient requirements. So am I concerned about the higher nutrient requirements? That I'm not entirely sure what's being asked there. That yes, as water is not the limiting factor, the plants would certainly have higher nutrient requirements. So that might enhance the benefits of intercropping because the nitrogen fixation from the pulse could help boost the non-pulse companion crop. And if water is not the limiting factor, they could take advantage of that extra nitrogen. It was probably a text question, so I don't know if the person can clarify what they're looking at, what they're asking, or they could try and contact me later. If you search my name online, you can find my email address and get a hold of me that way.
Awesome. Thank you so much, Michelle. Yes, I encourage if there's any additional questions to reach out to you. And thank you so much for your presentation this morning. It was fantastic.
Thank you. I'm happy to be invited.
All right, so next up we have Melissa Telfer. So Melissa has explored the mycorrhizal fungi as a potential disease break mechanism against chickpea ascochyta blight. She is a master's student with the University of Lethbridge in Agriculture and Agri-Food Canada. She's under the supervision of Dmytro Yevtushenko and Monica Gorzelak. She graduated with her BSc in biology from the University of Lethbridge in 2021 with a focus on plant physiology, molecular biology, microbiology, and parasitism. She worked for over 3 years during and after her studies in a plant pathology lab researching Fusarium pathogens in wheat. Her current project will explore the potential of a beneficial soil fungi, arbuscular mycorrhizal fungi, to be a disease suppression mechanism for chickpea blight. Take it away, Melissa.
Thank you, Mackenzie. Good morning, everyone. I'm pleased to be invited to speak today. My master's research is born out of the results that Michelle just shared about the chickpea flax intercropping and its effect on decreasing ascochyta blight. But I won't actually run my experiments until May in the greenhouse. So today, I'm going to present what goes on more underground with the arbuscular mycorrhizal fungi between them and your intercrops. So for the agenda today, I'm going to introduce what exactly arbuscular mycorrhizal fungi are, then discuss the hyposphere and common mycorrhizal networks, AMF's role in the nitrogen, phosphorus, and potassium nutrient cycles, how AMF contribute to facilitation and interspecific competition, sources of AMF and how to apply it if you're interested in using it, and arbuscular mycorrhizal fungi inhibitors, and any questions that you might have. So what is arbuscular mycorrhizal fungi? They are a soil-borne fungus that completely relies on its relationship with plant roots for survival while providing the plant with benefits itself. So then we say it's an obligate symbiont. AMF absorbs and transports nutrients and water from the far reaches of the soil that the plant roots cannot access due to distance or the small size of the soil pore spaces. It will do this in exchange for sugars in the form of photosynthetic carbon. This symbiotic relationship has occurred for over 400 million years with over 80% of land plants. Typically, it won't make this connection with the brassicas because the brassicas don't have the right mechanisms in their roots to support the symbiotic relationship. If you hear the term mycorrhiza, that's just the name that is given to the relationship between the AMF and plant root once successful root colonization has been made. So here I will show you the developmental process from an AMF spore germinating to the production of the arbuscule for which this organism is named. So first, the spore will germinate in the soil with the extension of the germ tube. The germ tube then branches, trying to locate a root. While this is happening, the root is exuding chemicals to attract the germ tube to it, and this is called chemotaxis, which means chemical movement. So let's zoom in on this section of the root where the germ tube has made contact. The germination tube attaches to the plant root cells through the creation of an aprosorium. Then a so-called infection peg extends from the aprosorium with enough pressure that it can actually puncture through mylar. Through a series of complex actions and chemical communication with the plant, it's allowed to penetrate the inner cortex cells of the root. And this is where the action takes place. The arbuscule is created, and this is the site of nutrient exchange. The arbuscules only last about a week before the plant disintegrates them, and the new ones are continuously formed along the length of the hyphae. But why this happens is still under investigation. The entire process from spore germination to arbuscular formation can take anywhere from 2 weeks to 6 months, depending on the amount and type of inoculum in your soil. So then we'll identify the different types of propagules, also known as inoculum sources for AMF. First are the spores, and I'm showing Rhizophagus irregularis because this is the species of AMF that is most common in managed agricultural land. They're very hardy and full of energy reserves, including fats, as seen in the blue multiphoton image photo. That's taken by Lee et al. In 2022. They survive winter in the soil and in root residues, which is one reason it's important to leave either living roots as cover crops or post-harvest roots if possible. Spores are the main source of inoculum, but like I mentioned, they can be slow to make the connection. In the bottom photo in B, we see a lot of spores after they've been sieved from the soil, and this is under a stereo microscope. And in C, we can see the spores in association with a root. They're the little circular structures. These are the intraradical hyphae. Intraradical means inside the root. There's also extraradical hyphae, which means outside the root. The hyphae are common to all fungi, but in AMF, there's no barriers along the hyphae to kind of divide it into sections. And the function of the hyphae is to absorb and transport the nutrients to deliver them to the arbuscule. And this picture I took, the intraradical hyphae are actually inside of a nitrogen fixing nodule of a pea plant. And this beautiful structure is called the arbuscule. They look like little trees, and that's how they get their name. And like I said, they're the ones that grow off the hyphae and last for only about a week. And as you can see, they increase the surface area significantly with all their little kind of tendril-like projections there for increasing nutrient exchange. And these are the vesicles They are the fungal energy storage organs. They're full of fatty acids and sugars. For some species of AMF, the vesicles can actually form new hyphae. So all of these parts together are termed propagules, and each one, depending on the species of AMF you have, can be used to grow new fungi. So it's not just reliant on the slow-growing spores. The fastest way is if you still have these propagules within your root systems, they'll form relationships in 2 to 3 weeks with your new plants. These propagules are also transported by macroorganisms in the soil, invertebrates, birds who are eating, and also long-distance migratory birds, because if they have ingested any root residues, the spores are excreted and then they can colonize new plants in distant places. So research is showing that AMF are vital to the health and defenses of crops. And after 400 million years, it's easy to see why. This is not an exhaustive list that I will present. And different species have different functional benefits depending on their environment and soil conditions that the plants and their AMF are specifically adapted to for your region. In southern Alberta, AMF are actually active whenever there's roots in the ground, so probably between the May to October. So AMF mobilize and transfer nutrients directly to the roots. They modify soil structure to increase the number of soil aggregates using a sticky substance, the name of which is under debate, but these aggregates are important because they consist of soil, air, water, and creates pore spaces in the soil. So the roots can actually access the pores that are full of electrolyte-rich water and oxygen. So without them, soil becomes compacted and dry, and it'll lack oxygen, turning anaerobic, leading to root decay, disease, and plant death. So this all prevents soil erosion as well by keeping the soil moist and intact. It also encourages the plant to produce more plant growth hormones, auxin, gibberellin, cytokinin, ethylene, and abscisic acid. And it's been shown to increase production of nitrogen by the legume plants in their nitrogen fixing nodules, which on the root here you can see are like the little round bulbs. They deliver phosphorus to the legume plant because they're working so hard to fix that atmospheric nitrogen. In legumes, it's called a tripartite symbiosis because you have the plant, the rhizobium bacteria, and the AMF. They've also been shown to suppress weeds, pests, and diseases through what's called mycorrhizal induced resistance, or the MIR. This is still being researched, but it's been shown to increase the speed and strength of defense responses in plants that are colonized by the AMF. Crops encounter a wide range of both biotic and abiotic stressors, as we all know. For chickpeas, because I'm studying them, the biotic stressors are living organisms like the animals that are eating them, nematodes, fungi, pathogenic ones, weeds, bacteria, aphids, cutworms. And the abiotic include non-living aspects like wind, heat, drought, flood, high temperatures, soil conditions, and nutrients. AMF can support the overall health and function of the crops by supplying water from the far reaches of the soil where the roots might not be able to access it. And this is especially true during drought. It supplies it directly to the plant roots. It supports the strong expression of genetics in the plant to combat stress. So typically are salicylic acid, jasmonic acid, or ethylene. And the emission, it can actually change the emission profiles of the plants through their volatile organic chemicals. And these are the chemicals that are released through the leaves and also underground through the roots. So if there's aphids eating the plant, it'll be like, hey, we don't want you here. Emit the chemicals that will attract the predators of the aphids, and they will come and eat them, and then the plant is protected. It also has been documented, as I mentioned earlier, with the faster defense response when AMF colonize the roots versus when they are not colonized. And of course, the increased nutrient supply, especially micronutrients and phosphorus, which help support the overall health and production of the plant and recovery when they experience these stressors. AMF have a thin layer of water on their surface that's in constant movement, and this is called the hyposphere. In this picture, I've highlighted it in the red box here, and the hyposphere is the light green area, and the hyphae then would be kind of the light blue to the left of the box. So in this hyposphere, the AMF transport microbes and other organisms around the soil. Considering the mycorrhizal fungi make up about 50% of a healthy soil microbiome, this is important. The hyposphere is as a source of water during drought and also bringing nutrient-filled microorganisms to protect and nourish the roots. The AMF can actually secrete specific chemicals to attract specific like bacteria and protists and things that will help protect the roots. So if you have like a pathogenic root fungi, then maybe some bacteria will come in and eat it, or there will be a beneficial fungus that it can also attract to the area, and that will kind of inhibit the growth of the pathogenic one. So arbuscular mycorrhizal fungi's claim to fame is their ability to form common mycorrhizal networks. This means that the hyphae of one germinated spore can colonize the roots of more than one plant. And it can do this with the same species. So if you're just doing a monocrop, this is still happening even in your intercrops as well. CMNs are highways for beneficial microbes through that hyposphere, nutrient transfers, and signaling molecules. Being connected to the CMN has been shown to lead to overyielding, as Michelle mentioned, especially in the intercrop system when the land equivalency ratio is shown to be greater than 1. The evidence for this is provided by labeling and tracking with carbon and nitrogen, as well as various tests of genes that are turned on or off compared to plants that are not connected to a CMN. The CMN also interacts, like the larger the CMN is, then the higher the rate of interaction with soil organisms like worms and beneficial nematodes and bacteria, which are all going to help to nourish and protect those roots. So nitrogen makes up 78% of the air that we breathe and therefore is available to plants. Legumes like chickpeas are able to fix their own nitrogen from the atmosphere into ammonia, which is plant available. In order for the plant to use it, they need special bacteria in the soil that make nodules on the plant roots, and those are called, the bacteria that do that are the rhizobium. One of arbuscular mycorrhizal fungi's job is to bring the nitrogen to the plant beyond the zone that the roots can access. And in the case, if you have an intercrop of, say, chickpea and flax, the flax is actually going to cause a stress to the chickpea, which is going to force it to fix more atmospheric nitrogen. And then the AMF, if they have a common mycorrhizal network, is going to transfer that nitrogen to the flax. Legumes can fix 200 to 500 kilograms per hectare, depending on which legume it is. And typically they don't need nitrogen fertilizer at all. As for the phosphorus cycle, 80% of phosphorus is not plant available. Most is present as phosphite, and the soil microbes convert this into phosphate. So your soil microbes are really important in this nutrient cycle. And high phosphorus concentrations can actually suppress the activity of the AMF. And plants that don't receive enough phosphorus from their AMF will actually sever the symbiosis. So like, you're not giving me what I need, you're out. To ensure the crops are still benefiting from AMF and not just the nutrients, like for the defense responses and such, it's important only to provide as much phosphorus as required. And AMF actually absorb and store the phosphorus until plants need it. So then it's not being leached, or you're losing it through runoff and erosion. And up to 80% of the fertilizer phosphorus requirements have been shown to be replaced by AMF in typical field conditions. In the potassium cycle, AMF has been shown to increase total K in plants by 20%, also ensuring less is lost to runoff and erosion. Potassium deficiency negatively affects root growth and shoot growth, and also AMF colonization, but still just only add what you need according to 4R for all your nutrient fertilizers. So facilitation is when one organism makes the local environment more suitable for another. Not unlike a human facilitator, plants can ameliorate the soil that results in pH changes, liberates nutrients in the rhizosphere, enhances the growth, survival, or reproduction of another plant. And this is plant specific, so in intercropping, be careful of the species that you plant together. But how do mycorrhizal fungi help? Well, the creation of that all-important common mycorrhizal network, the transfer of nutrients between crops, the lowering of the energetic expense for the plant in its production of defensive proteins, and they facilitate increased regrowth after damage or stress. They mobilize those microbes, nutrients, and signaling compounds from the soil and in between plants. And in intercrops, it's been reported through research that the presence of a pest or pathogen is actually signaled more efficiently when the plants are connected to the CMN. They think that this is because the root exudates might move slower through the soil, so the CMN will provide it a faster mechanism for the signaling to occur. The facilitation relationships can either be commensal, where one partner benefits and the other is just neutral, or mutual, where both benefit. Interspecific interactions refer to the interactions between different species sharing the soil space, so an intercrop, and even connected to the common mycorrhizal network. Legumes like chickpeas will rely on the AMF to supply them with phosphorus so that their nitrogen fixing nodules can perform properly. And as you can see there, the root hair shape and growth is actually positively altered in legumes by the AMF so that their nodulation rate is increased, and the nodule will actually adjust their shape to curve around the rhizobium bacteria, which makes it easier for them to form their nodules. They also increase soil stability through the creation of those important soil aggregates, and their hyphosphere is important in determining in supporting the crops that you plant. So the more diversity you have in your microbiome, then the healthier your plants will be. And this is especially important in times of disease outbreaks and drought. The entire system is so complex that, yeah, you can break down the importance of each individual organism in the soil, but it's really how they all work together that is the most benefit to your crops. AMF for the connection between the microbial community and your plants, but they also reduce species diversity, which in this case is good news because it will help control the weeds that you don't want growing in your intercrops. Overall, crucial ecosystem services are performed by all of our ecosystems for free, and this includes AMF. All the world's ecosystem services were given a price tag by the USA, Argentina, and the Netherlands at $33 trillion per year. If these age-old, naturally occurring symbiotic relationships are disturbed to the point where we have to start paying to support plant and soil health, growth, defenses, and resilience so that we can eat, then someone has to be willing to foot the bill. Instead, there are ways to maintain your soil health through methods like intercropping and supporting the microbial life, including arbuscular mycorrhizal fungi. If you're interested in using this in your production, you can actually use your own soil and increase your spores, other propagules on farm. I'm sorry, the pictures are a bit blurry on this one, but the one on the left is on farm, and they're just big canvas bags of farm soil, and then in there you would plant like a Sudan grass or some other host crop for AMF, and just leave it for the entire season. When it dies off, it'll create spores, and there will be other propagules that stay within those roots, and you can just use this kind of as like a fertilizer when you're planting your next crop. Cover crops are especially important. You can plant them according to what you need for nutrients for your next crop that you might be, if you're doing a rotation. You can till them under and leave them for 2 weeks before planting your cash crop. And studies have shown a 3 times increase in the number of AMF spores compared to just leaving your fields fallow and just letting the weeds take over. And there are commercial inoculums as well. There are mixed results on the viability and number of species that are in them. But if you have nothing and your soil is just completely dead, then this would be better than having no AMF in your soil, and you could give it a try. If you would like to use AMF, even just in your local gardens, it's still beneficial. There's different application methods depending on the location and size of the space that you have. In individual plants, you'll just put the inoculum straight in the hole. Larger gardens and fields, you can incorporate it into drip irrigation. In pots, you can mix it with your potting media. If you're starting with seeds, you can actually coat the seeds in the powder or granular inoculums. And trees and larger plants and shrubs and vines, especially grapes, love them. You can dip the roots into it before you plant them. Like I said, try what you need and see what works for you. And for fields, I would pick a smaller section and compare with similar sizes close by, you know, after a year or two, and see if you notice any positive differences for you. AMF are sensitive to disturbances, especially constant ones and over long periods of time. They are pretty hardy still. Fumigating your soil will destroy pathogens, but also the beneficial microbes. It's kind of like an antibiotic in humans. It might help initially, but there won't be any of the healthy soil microbiome left in order to protect from future pests and diseases. Deep tilling can destroy the mycelium by breaking it up, damaging spores, and destroying the hyphae. And compaction, we touched on this earlier, it removes the air spaces for the roots and fungi because they need oxygen as well. Fungicides, obviously it's a fungus, it's going to kill the AMF, so use sparingly and just as necessary. Over and misuse of fertilizers, especially the phosphorus, it'll prevent the association with the roots. And then of course, just environmentally will lead to leaching and runoff. If you have organic fertilizers on farm, those are best to use. The AMF will love it. And fallow fields, obviously if you have no living roots, you're not going to have AMF. And then the weeds take over, your nutrients leach away, and the wind, sun, and water will erode your soil, your topsoil. In this case, I would suggest using those cover crops. So in summary, we touched on how AMF develop, the important structures and their function, the benefits of having the association of AMF in your crops, abiotic and biotic stress support by the AMF, their hyposphere and what that does, the importance of the common mycorrhizal networks, AMF and the nutrient cycles, facilitation, the interspecific interactions, the inoculum sources, application methods, and inhibitors. I'd like to thank Agriculture Climate Solutions Funding through AFC for my funding for my project, and my supervisors, Dr. Monica Grizelak and Dr. Dmitro Yevtushenko, and my committee members, Dr. Michelle Hubbard and Robert Laird. And thank you for inviting me to speak today.
Thank you so much, Melissa. So we have a couple of questions that came in. The first one is, are there any specific forage crops that tend to produce more AMF than others?
There are, they absolutely love leeks. I don't know if anybody grows leeks. Wheat is a fairly good one as well. And flax, I found more AMF in the flax that I've been growing. I'm still waiting for it to colonize the chickpea, but that's part of my preliminary experimental process. And Sudan grass, but I don't think you'd actually grow that as a crop around here.
Awesome, thank you. The next question is, if flax is benefiting from the nitrogen from wrapping up the P N fixation, is it giving carbon to the P through the AMF, or is it stealing carbon from the P?
I don't think that it's stealing carbon from it. In this symbiotic relationship, the plant is giving carbon to the AMF so that it can run its life processes, right? But in exchange, it's bringing all those extra nutrients and providing some signaling through the pathways for defense responses and things from the other plants that are around it that might be experiencing stress. And so even if it is taking a little bit more carbon, like the plant isn't going to allow it to take without giving back. So the plants have to be receiving some sort of equal or greater benefit in order for the carbon to be delivered to the AMF. So I don't think that it could really steal in that sense.
Awesome, sounds good. All right, the next question is, would biofumigants like the full season mustards mowed and tilled in ahead of potatoes have the same detrimental effects as commercial fumigants?
I am not sure how to answer that because I don't farm myself either. So I think if it's fumigant in a local area versus like, I don't know if it's just going in with the potatoes, etc., or if it's like the entire field that's being affected. If it's more of a local environment, then you'll just kill whatever's there. I don't know the half-life of the fumigants either. So if it's short enough, then maybe the microbes will come back and recolonize. But I would really just recommend supporting the creation of the microbiome that can fight off whatever pests or pathogens you're having issues with, rather than just like killing everything, even if it is just a local area or whatnot. Hope that helps.
Awesome, thank you so much. All right, our next and last question is, what impact would tillage radish have on AMF in a field?
Ooh, I love tillage radish, especially before potato crops. That one, I haven't come across any research yet specifically for AMF, but it could create the soil conditions that would allow AMF to come in and colonize, and it would provide it like the overwintering residence, if you will, for the spores and the propagules, so that as I understand it, with the tillage radish, you leave it there to kind of rot before you plant your new spring crop, right? You don't want to take it out because then you lose all those nutrients. And so the AMF will be in there so that when you plant your new crop, then your propagules are going to be there. They won't just be the slow spores. And so within 2 to 3 weeks, they'll help your new plants to be better established and already have a healthy microbiome to fight off all of those pests and pathogens that might be coming around.
Awesome, thank you so much, Melissa. And again, thank you very much for coming this morning, presenting that wonderful presentation.
Thank you.
All right, everyone, next up we have Dr. Sean Sharp, who will be speaking on his evaluation of integrated weed management strategies for camelina and field pea intercrops. Dr. Sharp is a research research scientist with Agriculture and Agri-Food Canada at the Saskatchewan Research and Development Centre in Saskatoon. Dr. Sharp received his BSc from Mount Allison University in Saskville, New Brunswick, his MSc from Dalhousie University, and his doctorate from the University of Florida. Dr. Sharp continued at the U of F as a postdoc, working on developing artificial intelligence to detect weeds for a precision sprayer, herbicide efficacy and safety trials, and studying weed ecology. Dr. Sharp joined the Saskatchewan Research and Development Centre in Saskatoon, Saskatchewan in 2019. Dr. Sharp works involves preventing and mitigating herbicide-resistant weeds such as kochia and wild oat in field crop. Thanks for coming this morning, Dr. Sharp, and you can take it away whenever you like.
Thank you so much, Mackenzie. Excuse me. And good morning, everyone. It's great to be able to be here today to chat to you about weeds. They are a very important part of my life, and as far as research goes, my first interaction with intercropping was actually while I was in the United States doing my postdoc. And in strawberry production there, imagine rows of black plastic mulch and palm trees and sunshine. There they grow their strawberries on their mulch, and they would actually come in later, about halfway through the season, and put cucurbits in so that they could extend out their use of that mulch and increase their growing season. So when I moved here and I was getting to understand the high priority weed targets like kochia and wild oat, which we'll talk about just very briefly. Intercropping, of course, came back into interest as a way to kind of help manage these weeds. So kochia is one of the major weeds which I work on, and it's an early emerging annual with a long growing season, and how it's kind of come into my lab is because of the herbicide resistance that is evolving in this species. We have resistance to groups 2, 4, and 9, and these genes can be transported in pollen as well as transported via tumbling diaspore, also known as a tumbleweed, that's going to move around in the wind, and these are quite prolific seed producers. The 4100 is probably a bit of a lowball estimate, but yeah. So interesting about their ecology is that they only have a very short seed bank persistence of about 3 years. So aggressive management can help drive down infestations. The other target that is on top of mind for me is wild oat. And again, this is another spring annual, but contrasting with kochia, wild oat has a very long emergence window. So with kochia, it's going to be up very early. It may come up later in the season as well, but the bulk of it's going to be early emerging, whereas with wild oats coming up later in the season. So the two of them kind of have an emergence profile that's conflicting. So it's hard to manage both at the same time. And again, it has resistance to groups 1, 2, and formula 8, which is triolate that's been regrouped into group 15, which is the same mode of action as peroxasulfone, which is often tank mixed in with group 14 pre-emergence residuals. So herbicide resistance risk is an important element of my work. And today, this here is just an overall framework that talks about different BMPs to help either prevent the establishment of herbicide resistance in your weed populations, or to help manage those populations if they're already in your field. And the premise is basically that we want to reduce the soil seed bank. We want to have it static or declining over time. We want to use our understanding of those weeds and to scout to try to find them. We want to use all available control measures. We want to take pressure off of our herbicides. The issue is that they're they're breaking faster than we can produce new modes of action with these weeds. So we want to take pressure off of them. Primarily with what I'm going to talk about today is going to be cultural practices. And this is where my interest in intercropping really starts to evolve. Because intercropping fits very well in with this BMP7, as well as using increased seeding rates to try to achieve canopy closure. And we'll talk about that a little more. And the other BMP that we're going to evaluate today, or not evaluate, but kind of touch on today is going to be BMP5, which is to be used multiple modes or mechanisms of action. And so this is using different herbicides to kill plants in different ways effectively. So we want to use a different type of way to kill it so that we're having to kill fewer weeds, because with resistance risk, every weed that you kill with a herbicide has risk. So we want to basically lower down all of the population so that when we're spraying, there's less weeds to kill at any given time. So the two crops of most interest in these studies is going to be yellow field pea, and we're using CDC Amarillo for this, which is a fairly standard pea grown. And then the species of interest for the intercrop is camelina. This is also known as false flax, gold of pleasure, among many others. It's been cultivated and domesticated in Eurasia. What's interesting about camelina is that it has a good pest profile. While it is a mustard, it has good tolerance to pests that are plaguing canola, such as flea beetles, as well as it's been noted as a very good weed competitor. So its tribe, or the group of plants that it's most closely associated with, is not canola, but it's actually Arabidopsis, which is a model plant that's very well known in the academic world, as well as shepherd's purse. So it grows a lot like that, only it's bigger typically, but the overall growth form is quite similar. Pardon me. So in this research, we will be using a group 3 herbicide, which are mitotic inhibitors. So I just want to run through them a little bit first. So ethofluralin, or EDGE, is one of the two that are available to use in or on the prairies, as well as trifluralin. Ethofluralin is the product that we'll be using today. So these are applied to the soil. They're typically a granular herbicide, and they're absorbed by seedling roots and shoots. So once those seeds germinate, and in our no-till systems, most of the weed seed is going to be near the top of the soil profile. It's going to create a barrier. So these herbicides are not very mobile in water. So they're not really going to move around in the water, absorbed with the water. So they're just going to create a barrier that those weeds are kind of stuck in. Whereas we typically plant our crops below that. They have resources down there, and then they can punch through the soil to get above ground. So what we see typically for control is actually that weeds do not emerge. And it's very immediate. So ethofluralin is used in no-till systems. It can be applied in the spring or the fall. Again, it is a granular. And with the no-till, usually you'd use a harrow to incorporate it, so you get a very narrow protection or a wall in the top of your soil to kind of keep those weeds down. And then, of course, especially for wild oat, where it's a larger seeded spring annual, and its seeds do have the ability to drill into the soil some as they're falling. They do recommend pre-seed burn off with glyphosate to control any weeds that emerge, because with this herbicide, if they're already emerged, not going to work. So just a few considerations about ethofluralin into production. In the spring, you want to apply it 10 days prior to seeding, or in the fall, you can go October 1st to freeze up. Its persistence is linked to moisture, although what I do like about this product and this mode of action is that moisture is not need it for it to work. So unlike the group 14s, you may lose efficacy if you don't get that moisture. And if you're not in an area with irrigation, you might not have a lot of options to make that herbicide work. Whereas with group 3, you're not reliant on that particular need from the environment. There is some caution. Wheat is a very important crop on the prairies, and if you are, for example, doing a canola wheat pulse rotation and you put it in your canola and you put it in your pulse, you're likely going to injure your wheat. So only one time within, so 2 times prior to wheat is unadvisable, and that's on the label, of course. So with pre-emergence herbicides, they're very excellent at controlling weeds while they're emerging, unlike there are post-emergence herbicides which require the weed to be present and there to be tissue present above ground that we spray. So their efficacy will wane, and what we want to do is build a cropping system that's going to be able to close canopy rapidly. It's going to be very competitive for light and space particularly, and that way there's less light and for the weeds to grow. There's going to be less ability to fix carbon and steal the resources that we're intending for the crop. There's lots of ways to encourage a crop to close canopy. I've got a couple of them listed here: row spacing, the rates of seeding, of course, as well as developing intercropping. You could also use cover crops, which were mentioned earlier. They can help with that. And this is important because another weed that I'm very concerned about that we don't have yet in Saskatchewan, it's starting to see it in Manitoba, is water hemp, as well as threats from Palmer amaranth moving from the north. So we want to try to encourage and build resilient cropping systems that are very competitive, so that if a seed does manage to move up in contaminated grain, or if it's moving up in waterfowl, for example, in the spring migration, or that our systems will be able to handle that, and they'll be able to compete and smother out those weeds as those threats are moving into our systems, and which they already are happening. So as I mentioned, cultural practices are important in BMP. This is actually some work done by Steve Shirleff's group looking at peas and their ability to suppress weeds, looking at different rates of seeding. The standard is kind of here in the middle. And we have our increased rates of seeding here, biomass production. So the weeds are these black boxes, and then our crop here is the white boxes. And that you can see that increasing the rates of seeding just as a pure monoculture does have some benefits. So peas' ability to tolerate intercropping should be fairly well, and as well. So we did some work last summer in Saskatoon at our research station here. The first trial I'm going to talk about is dealing with peas and camelina intercropping. So we did ethylfluralin, yes or no, as well as 3 rates for camelina seeding. We were targeting 300, 550, and 800 plants per square meter. Put this out in the spring using an Aerotech plot seeder. We did 6 rows of the camelina, and then we would move over. So that was at a 12-inch row spacing. We move over 6-inch and then put down another 6 rows of the pulse. Actually, the opposite order. We put the peas in first, sorry, and then we move over and put the pulse in typically. So this is a 2-factor factorial. So we're looking at the effect of the herbicide, and then we're looking at the effect of the camelina rates. And then I did analysis in SAS for just a standard ANOVA and Tukey's on a significant difference test. So this is just an image. This was from August. And at this point, the season's fairly well in, just for reference. So the camelina stand density was not responsive to treatments, either from the herbicide or from how much seed we're putting in the ground. It was very dry in Saskatoon last year. So I think with the limited amount of moisture, it lowered the carrying capacity of those fields lower than then we would see responses and rates, unfortunately. So with the pea stand, we did see an effect of the herbicide as well as an interaction with Camelina seeding rates. I just want to point this at the top, which is the 29 plants per square meter, and where we treat it with ethylfluoralin and then untreat it with 40. This is a case of do what I say, not as I do. We did plant one day after treatment. There was a bit of a miscommunication with the team in the field, so they did treat a little further, and they did. Instead of harrowing, they did disc it in probably about 6 inches. So I think that injury is likely due to that and not necessarily the fault of the herbicide. It was more likely how we put it in the ground. Whereas the untreated plants reached about 40 plants per meter squared, we were targeting 80, so it's still a bit down. And then this interaction, there's not a whole lot to see here. It's driven largely by the effect of the herbicide. Here you can see that the number or the letters all have C. There's not really any differences, whereas with the untreated, we start to see some differences. But yeah, largely it's the effect of the herbicide that's affecting those stand densities. Just a few images here. Again, this is that later timing in August. So you can see here the peas, and this is the peas intercropped with the camelina. The camelina definitely dominated the stand. You can see some of the peas kind of snuck out at the top of the plot since there was some light availability, and there's a little bit kind of in here, but largely it's camelina that you're seeing in that image. So I talked about canopy closure. So to measure canopy closure, I had 2 long light bars, and I would put one under the canopy, and I would hold one above the canopy, and then I would have someone take a reading in each plot. And this would tell us how much light was actually being intercepted by that canopy. So this is directly back to closing it off and not giving the weeds light to grow. So we did this twice, once in July. So we seeded early June. So this is about a month and a half after planting, and then 2.5 months in August. So by August, things had already closed up. So there's not much to report. So I'm just going to focus on what happened in July. This was the earlier timing. And in my mind, this is the more important one since we want to create these differences earlier in the season. So again, there was an effect of rate, no effect of the herbicide, and then just a general trend. So when the peas were in monoculture, they only absorbed about 32% of that available light that was coming in, whereas when we started adding camelina, the canopy reached 70% capture, and that was about the max that I was seeing even in August. So very good news with the camelina, with the pulses, we're able to close that canopy quicker, and that should help with weed suppression once the ethylfluoralin would start to wane. So weed density effects. Weed densities at the site were fairly low. I was actually hoping for a little more of a weed competition. Just at the next trial over, I had some wheat and barley, and it was swimming in wild oats, so I don't quite know what happened there. So next year we're going to encourage it a bit more and seed some kochia and wild oat into this. But for now, we had about 4 plants per meter square in the untreated, and then this is for mid-July, sorry, and then about 1 plant per meter squared where we applied ethylfluoralin. So that's still a fairly good reduction in how much weed biomass and is there to compete with the crop. At the early timing, no effect of camelina density, whereas when we reach the mid-August timing, we see the effect of the herbicide and the density of camelina. There's no interaction for this one, so fairly straightforward. The monoculture peas had 4 plants per meter squared for weeds, and this is across all species. It's quite variable, so I wasn't able to pull out any specific species, unfortunately. Whereas with the camelina and peas, we did get it down to 1. So that's a considerable drop based on the camelina peas, and I would anticipate this is due to the light effects and that canopy closure that we were seeing with that camelina earlier in the season. Whereas with the ethylfluoralin main effect, the untreated plots typically had about 3 plants per meter squared, whereas they reached around 0. So excellent control with the ethafluralin in August. For yield effects, camelina was not responsive to treatments, which we had anticipated since the stand densities hadn't really responded as well. So the average yield is about 97 grams per meter squared, and then the 1,000 kernel weight is about 1.5. So nothing too exciting for effects from the pulses. Camelina is definitely the dominant competitor here in this matchup, and we'll see here the P was definitely not. So here we have the camelina density and that we were targeting, and then we have our P yields. And so this is the monocrop here of P since we don't have any camelina. We can see we've got 104 grams per square meter of grain yield, and this drops to about 30. And then there's actually a bit of a drop again with the 800. So there is some yield effects, and then we do see a bit of quality drop with this competition with the camelina as well. So there may be some implications there. All right, so the second study I want to talk about, this was actually suggested by Dr. Christina Ike, a collaborator on this project. She's the camelina breeder, as well as other alternative oil seeds here at SRDC in Saskatoon. And she was interested in other species and how they might compete with camelina. So we worked with 5 different species: lentils, fabas, chickpeas, peas again, as well as black dry beans, and we just use the standard 100% target that SaskPulse was recommending, and then we grow them either with or without camelina at 550 plants per square meter target densities. And we had a control as well where there was no camelina, or where there was just camelina, sorry. And again, this is put out with the Artex 12-inch row spacing. We'd put the pulse in first, move over half an inch, or sorry, yeah, 6 inches, and then feed the camelina then. So we did see that the pulse densities were influenced by species choice. So these were the results and densities we were getting. Peas were fairly similar to the last trial. The lentils were quite a bit lower, but about half, and then the chickpeas and the dry beans were less established. And again, part of this might be due to the way that ethafluralin was incorporated. This trial had ethafluralin all across it, so we weren't actually interested too much in the weeds. It was more about the crop competition dynamics in this one. So the camelina was not, did not impact the pulse 1,000 kernel weight in this one, which was good to see. We did see some quality estimate or quality reductions with the peas and the camelina in the last study. This one, we're not seeing that here, but we do see that drop again. We got chickpeas at the top. This is the monocrop. So this is just the peas or the other pulse, and this is the intercrop with the camelina. And then you can see here, these letters you can compare here, but don't compare these ones. These are only comparable within. So these ones you can compare across, and you can see that there is reductions, and the degree of reductions does depend on the species involved. So with lentil and dry bean, they were very poor at competing with camelina and still being able to produce good yield. The fabas, you'll notice, aren't on the screen. We had some poor establishment. I think it was probably injury from the drill, so we're going to evaluate some other options there. It's just a few images of this as well. So this is the chickpea. See here, pretty good canopy closure, not really seeing too much chickpea in there. There's the dry beans. Got looks like wild oat at the top there. You can see a few of the dry beans peeking in the bottom there. They're well below the canopy. The lentils, green foxtail in the front of the plot there. Good stand establishment it looked like, even though the stand numbers weren't quite where we were looking. And then again, You can't even really see too much of the lentils underneath the camelina there. So to wrap up, I just want to acknowledge that this research was co-funded with Gowan Canada, who also provided the herbicide, the edge, ethylfluralin, as well as the Saskatchewan Ministry of Agriculture through the ADF program. And I want to, of course, acknowledge my team of technicians and FSWEP students who put in a lot of long days that they're helping to get the data off these trials. So I will wrap up there, and if there's time, I'm happy to field any questions.
Awesome. Thank you so much for that great presentation, Sean. It doesn't look like we have much time for questions, but that was a great presentation. It was super interesting and super informative. Thank you very much.
My pleasure, of course. Thank you for inviting me.
Absolutely. All right, everyone. Next up, we have Brody Erb. Brody will be speaking about intercropping flax with soybeans and field peas. Brody grew up on a family farm just outside of Winnipeg, Manitoba. He completed a diploma and undergrad in agronomy at the University of Manitoba, and currently he is a technician for Kirsten McMillan at the University of Manitoba. He works in the soybean and pulse agronomy lab and is also completing an MSc related to field pea agronomy practices. Take it away, Brody.
Alrighty, thank you. Okay, good morning, and thank you for having me to present some of our intercrop findings from our research here in Manitoba. I'm Brody Erb. I've been a research technician for Kristen McMillan's Applied Soybean and Pulse Agronomy Lab at the University of Manitoba, going into my 4th year. I'm also currently studying the impact of several agronomic management strategies on pea health and ultimately yield as part of an MSc. Given my background, a greater focus of this presentation will be on the applied agronomy aspects of our research on these intercrops to date. So why intercrop? Well, I think Michelle covered that fairly well. But to restate the primary objectives that we have for optimizing efficient and sustainable cropping systems using the intercrop as a tool, improving profitability, which we analyze by means of marginal profit, over yield, which we represent as land equivalent ratio or LER, system stability, and efficient resource use. Here in Manitoba, our provincial intercrop success story is pea canola, which has been demonstrated to consistently produce LERs which surpass its monocropped counterpart. It is goal of our research lab to enhance the limited research for the intercrops, which I'll be presenting at the local level. Our 2 sites have been Arborg, which is north of Winnipeg, and Carman, which is southwest. Both sites received higher than average heat except in Arborg's first year. Precipitation, on the other hand, was considerably lower than average for both sites. In 2021, Arborg was assessed an exceptional drought designation, which is a 50+ year event, and you'll soon see the yields are a reflection of that. From one extreme to the next, in 2022 in Arborg, that was the exception to the lack of moisture, and in fact, overland flooding was a significant issue in the area, especially early in the season. As I mentioned, our lab is studying several intercrops and at multiple seeding rates. These include soybean flax, which is seeded in alternating separate rows, pea canola, pea flax, and pea oat, which are mixed solid seeded. Getting into more detail on the soybean flax and pea flax intercrops, these were the seeding treatment parameters. Each intercrop had 3 seeding density levels. Otherwise, they were treated the same. Since soybeans and flax were seeded on separate rows, we were able to seed at optimal depths. For pea flax, seeding depth of 1 inch was used. Each intercrop only had starter application of phosphorus at 15 pounds per acre, which was seed placed. One of the largest challenges for these intercrops is the limited in-crop herbicide options. Each treatment received a pre-emergent application of Authority 480. The soybean flax intercrop had 2 in-crop applications, Post Ultra and Basagran Forte. These 2 products are not registered tank mixes and therefore had to be applied separately at recommended 4 days apart. Pea flax had an in-crop application of Post Ultra, but MCPA is also registered for both crops. Here are some of the images from both sites of the soybean flax intercrop to demonstrate the row configuration, but more importantly, the tolerance of the system for abundant weed communities. In Arborg on the left, plots are weed-free as there was good herbicide control. That was not the case in Carmen, where there is a high population of Group 1 resistant green foxtail. As such, controlling this weed was problematic for both years at this site since no in-crop herbicide options are available to combat it. A rigorous and comprehensive weed management strategy is required to grow this intercrop under these conditions. It is therefore recommended that one should take great care to select sites with low weed seed banks and no history of herbicide resistance. Maturity of either crop in the system varied greatly depending on the environment. In the extremely dry year of 2021, flax matured more than 20 days ahead of soybeans due to early senescence of the flax, as well as a late season rainfall, which prolonged the soybeans. Under normal conditions, in 2022, the crops matured more evenly. Desiccation in both years made threshing much easier and more punctual. Here's an image of each of the 3 soybean flax intercrop seeding rate levels. From left to right, soybean at a full rate of its monocrop counterpart, flax at 3/4 rate. Next is soybeans at 2/3 rate and flax at half rate. And last is the soybeans at 2/3 rate and flax also seeded at 2/3 rate. Visually, you can see the difference in proportion of seed in each sample. Of note is the high dockage level. This was a result of attempting to thresh fully the flax bolls and thus limit harvest loss while avoiding cracking or splitting the soybean seed. Cracked soybean seed is problematic in that it not only downgrades the quality of the seed, but cracked and split soybean can be very difficult to separate from flax while cleaning. This can lead to flax impurities, which can be costly. Increasing the fan speed may have improved the threshed sample by lowering the dockage. However, this was not fully explored. Instead, dockage and foreign material was reduced during the seed separation process. Here's a look at the soybean flax intercrop LER and marginal profit results in Arborg for the 4 site years. I will highlight a few of the interesting findings. Soybeans in either cropping system, monocrop or intercrop, performed poorly in all years. Flax, on the other hand, had good yields in both systems and had greater contribution to LER in the intercrop system. Intercropping tended to underyield compared to its monocrop counterparts, with a 4-year average of 0.93. At a score of 1, the accumulation of both crops in their intercrop equaled the yield relative to the monocropped counterparts. A score of over 1 would indicate overyielding, which is of course a good thing. A soybean flax intercrop tended to be more profitable than monocrop soybeans, but less profitable than monocrop flax. Compared to all other treatments assessed in the 4 years in Arborg, both monocrop and intercrop soybean flax usually had a below average marginal profit rank. Of the 3 different seeding densities, seeding soybeans at 2/3 rate and flax at half rate tended to be the most economical due to a combination of equal yields to other treatments, but at a reduced seed cost. Before I flip to the next slide, you'll notice that in 2021, there was the aforementioned extreme drought this year, which resulted in crop failure. I decided to separate the locations when presenting this data because of some of the extreme environmental factors that we experienced while conducting the experiments. In Carmen, most notably, as I said, was the grassy weed populations. This unfortunately caused all flax treatments to essentially fail. Here, the intercrop LER was also less than 1. Intercropping soybean and flax was more profitable than flax monocrop, but much less profitable than the monocrop soybeans. So what were some of the takeaways from the soybean flax intercrop? We found that soybean and flax should be grown on alternating separate rows to prevent overcompetition of flax onto soybean. Soybean at 2/3 rate and flax at half rate tended to be the most profitable of the levels that were studied. Variety selection is key to manage or maintain equal maturity between the soybean and flax to limit harvest loss and seed degradation. We used a very early season maturing soybean variety, NSC Watson and NSC Dauphin in Arborg, and a mid-season maturing S007Y4 in Carmen, paired with CDC Glass, which is a later maturing flax variety. We only applied starter phosphorus to the intercrop treatments. There may be room for further investigation for more optimal fertility strategy. Weeds are difficult to control in this system, especially grasses, and even more so if they have group 1 resistance. Site selection with low weed seed bank is critical. Pre-seed or pre-emerge applications are also likely important, as is early in-crop application. Harvesting soybean and flax requires fine-tuning thresher specs for best results and limited harvest loss. Use of a harvest aid is likely very helpful to both the maturity of both crops, but also to improve threshing by way of complete plant desiccation. And now on to the pea flax treatments. Pea aphid data has been collected at all site years since 2020. Currently, we have only analyzed the first year of collection. In this year, pea flax intercrop reduced the number of aphids observed per plant compared to the pea monocrop. Whereas soybean flax widely ranged in maturity between either crop, pea flax is typically matured more evenly and with a narrower range. This system was also impacted by the 2021 drought, as demonstrated by the pictures on the right. Like soybean flax, the size of pea and flax bolls are also very similar. Since peas are slightly larger in size than soybeans, the harvest of this intercrop can be even more challenging. Prevention of pea seed cracking while obtaining a full thresh of the flax bolls requires fine-tuning of combine settings and inevitable damage to some pea seed or loss of some flax or both. Increasing fan speed may also lower dockage than our settings. However, we erred on the side of caution so as to not lose flax seed out of the back of the combine. Unthreshed flax bolls in composite sample cannot be separated by size, but rather by air or gravity table. This will unsurprisingly lead to flax seed loss, so it is important that they are properly threshed. Using a desiccant prior to harvest may avoid or limit these complications. On the land equivalency ratios and marginal profit results for the pea flax intercrops we tested. In Arborg, both pea and flax grew well in both monocrop and intercrop systems, except for 2021. LER for the intercrop tended to be close to 1. Both monocrops tended to be quite profitable and more so than the pea flax intercrop. Seeding combinations with a higher proportion of pea and lower proportions of flax tended to have a lower LER, but were on average more profitable. Of all the systems tested in this experiment, the pea flax treatments usually ranked middle of the pack in terms of profitability. Flax yield again was hampered by weed seed at the Carmen site. The low monocrop flax yield led to deceivingly high LER for the intercrop. Because of the failed flax crops, the marginal profit for the intercrop tended to be greater than that for the monocrop flax, but it was significantly less than the pea monocrop profit. The takeaways for the pea flax intercrop were pea flax performed well in mixed solid seeded systems. Seeding pea at full rate and flax at quarter rate tended to retrieve the best marginal profit of the 3 levels tested. Variety selection is a great way to narrow maturity dates between crops and improve harvest. We used CDC Amarillo as well as AAC Chrome peas, partnered with an earlier than Czech maturing flax variety in CDC Plava. The fertility program was the same for all pea flax treatments as well as soy flax treatments, a 15 pound per acre seed placed phosphorus application. More research could be done to optimize economic returns and resource use efficiency of fertilizer applications in this system. Weed management. Resistant grassy weeds and some prominent broadleaf weeds like wild buckwheat and redroot pigweed can be devastating to this system. Site selection to avoid these weed populations is very important, as well as pre-emerge or pre-seed application. Harvest management. Minimizing flaxseed loss while also minimizing pea seed coat damage can be a challenge. A pre-harvest desiccant will likely help. And that concludes the presentation. I just want to thank MPSG and Prairie's East Sustainable Agriculture Initiative. And yeah, I'll take any questions if there are any.
Awesome. Thank you so much, Brody.
No problem.
So the first question that we have is, does the pea flax monocrop lower the amount of disease found in the pea crop?
So like I kind of showed in some of the slides, we had pretty dry years with the exception of the last year in Arborg. So there wasn't much disease present, and we actually didn't really measure disease. So I can't really answer that.
Fair enough. The next question is, do you think that the soybean flax rate would change if you had more ideal conditions?
Yeah, probably. It was unfortunate. Like I kind of wish we had seen maybe the best of both sites. A lack of or a lower weed seed bank with less resistance, but also some more rain. So I think under ideal conditions, that intercrop may have improved with different rates as well.
Awesome, thank you. And the last question here is, did you account for the split peas that you were talking about on harvested grain? Did you account for those split peas when you were calculating your marginal profits?
we analyzed these split peas in our sample, pea sample and pea grading. So yes, I do believe they were contributed to the marginal profit calculations.
Fantastic. Thank you so much. Let me just check if we have any more questions here from the audience. All right, Brody, thank you so much for your time today and for the wonderful presentation.
No
We
problem.
have no
Thanks
more
for having me.
questions from the audience, so have a great day.
Thanks, you too.
All right, everyone. So the lastly, we have two members from SCIC, Arlen Frick and Dale Naus. Arlen is a senior analyst in the Research and Development Unit at the SAS Crop Insurance Corporation. He works on policy and program development for organic crop insurance, weather-based programs, and intercropping. Arlen has an MSc in soil science and farmed with his wife in Melville for 20 years. Dale is the manager of strategic operations and process improvement for SCIC's insurance unit and has worked with SCIC for almost 20 years. Prior to SCIC, Dale worked as a representative for several egg cam manufacturers and egg retail. Dale is passionate about agriculture, and throughout his career, he has focused on building positive relationships with agricultural producers and industry representation. Dale engages with the industry and its customers as SCIC adopts new products, offerings, and technology, and is excited to see the benefits flow through the SCIC customers. Dale enjoys working closely with SCIC staff, customers, and industry partners with the goal of contributing to everyone's mutual success. Take it away whenever you're ready.
Hi, can you hear me now?
We can hear you now, thank you.
Okay, is my screen showing the way it's supposed to be?
You bet, it looks great. We also
Okay,
see your pointer.
Okay, good. Okay, so I'm going to be talking about crop insurance and how intercrops can currently be insured under our diversification option. So our definition of intercropping, or particularly for grain intercropping, which most of the speakers have been talking about today, is the practice of seeding multiple crop types on the same land with the intent of separating and marketing individually. So that's kind of our official formal definition for intercropping. And so most of the time when people think about intercropping or say it, they're mostly referring to grain intercropping, and like most of the speakers are talking about today. So that's what I'll be talking about mostly, and that's insurable under our diversification option, and that's been kind of the same process since 2018. And any intercrop mix is insurable under that. So I'll mostly be talking about that, but I just wanted to also quickly mention some other types of intercropping that are insurable. So one's kind of forage intercropping, or sometimes referred to as forage polycrops. So basically a multi-species mix, intended to be harvested as a forage. And so if it's predominantly an insurable cereal, so a cereal that's insurable under our green feed program. So, I mean, the common ones would be oats, barley, triticale, things like that. It can be insured under our green feed program. So that's, again, if it's predominantly that cereal. So it could be insured as green feed oats, green feed barley, for example. But if it's a, not predominantly one of those cereals, if it's a 5 or 10 species mix of other species, that could be insured under our forage diversification option. And so I'm not going to talk about that one in detail, but you could contact one of our offices if you need more details on that one. So basically any mix can be insured under that. And it's a proxy program, so it's based on area barley yield. So another thing that's sometimes referred to as intercropping is basically underseeding forage crop, or relay cropping could also be referred to as that. So that's been going on for decades, obviously, or maybe even longer, that farmers have used that. So if it's an underseeded crop that's got a cover crop, it could be insured for whatever its intended end use is. If it's intended to harvest for grain, or probably more commonly green feed, so it could be insured as that if all the other insurance requirements are met. Not sure what just happened. Let's try this again. Okay, I think I'm back. And the underseeded forage crop can be insured for establishment. And so most common forage types can be insured for establishment. So alfalfa grasses, alfalfa grass mix, sweet clover, for example. So the diversification option, which again I'm going to talk about mostly, it's not just for intercropping, although that's how intercropping is insured, but any other kind of specialty crop that we don't have enough data that we developed a yield loss program for. So basically not insurable under our kind of core usual multi-peril crop insurance that all the common grain crops would be insured under. So not just intercropping, it could be anything like borage, quinoa, spelt, any kind of specialty crop where we don't have a yield loss program. So to be eligible for that option, you have to have kind of insured acres of the traditional crops, so grains, oilseed, pulses under that multi-peril program, and a maximum of 30% of the acres of those traditional grains. A maximum of 30% of those acres can be insured under diversification. And I'll go through an example to kind of illustrate that too. So as far as intercropping, any intercrop combination, any crop mix can be insured, as I mentioned. And the diversification option is a proxy program. So it's not based on the diversification crop itself, but on the other multi-peril crops that are insured. So the dollar per acre premium and claim are based on the average of those other crops. So it's really any claim will reflect kind of the average on-farm crop performance. So, you know, if there's an area drought or frost that affects all your other crops, that will be applied to the diversification crop as well. So now an example. So in this example, your other kind of typical or traditional multi-peril crops that are insured are spring wheat and canola. So we've got 1,000 acres of each. This is just, you know, a hypothetical dollar coverage that would be applied to each, the premium for each. And so basically we use the average of those for diversification. So the average coverage would be $400 an acre, average premium $12.20 an acre. And in this example, it's got a small claim or, you know, medium-sized claim on both of them for a total of $300,000. So over the 2,000 acres, that's an average of $150 an acre. So those average numbers are what's then applied to diversification. So just a quick aside, I said there was the 30% acre limit. So in this case, got 2,000 acres of multi-peril crops. So 30% of that is 600 acres. So you can insure up to 600 acres under the diversification option. So continuing the example, so those are the average that I was talking about for the multi-peril crops, the $12.20 and $150 acre claim, $12.20 premium. So in this example, the diversification crops, they have two different ones, quinoa and an oat pea intercrop. So 100 acres of quinoa, 400 acres of oat pea. So 500 acres total, so you're under the 600 acre limit. So those could all be insured under diversification. The coverage, the $400 an acre. So that's really what represents the maximum claim you could have. So if you had a complete loss under the hard red spring wheat and canola, you could potentially get $400 an acre for the diversification crop. So the average premium of $12.20 an acre for those multi-peril crops is what's applied to every example, so both of them get the same $1 an acre premium. And then the claim, again, was $150 an acre on average. So that would be also applied to those diversification crops. So again, it's a proxy, so it's not based on the actual yield of the quinoa or the oat pea intercrop. It's based on the average of the other crops. So whether those diversification crops are, bumper crop successes, or, for whatever reason, a complete failure, the $150 an acre would be the claim or the claim generated for both of them. And this is just multiplying it through by the acres, and that would be the total claim or indemnity you'd receive in that case. So it just shifting gears a little bit past that example to kind of some of our stats that we've collected at crop insurance for intercropping. So this is the last 5 years, so starting in 2018 when we started insuring intercrops under the Grains Diversification Program. And this is the acres report to us of both insured and uninsured. So insured acres would be under that diversification program. Uninsured would just be reported to crop insurance by our customers, but not insured under that diversification. So you can see that kind of acres peaked in 2019 and have tapered off a little bit since, but I would say it's kind of stabilizing, kind of between 20 and 30,000 acres reported to us a year. So the next slide is kind of some of the common crop mixes that are reported to us. And so these will be, probably not surprise anyone. It actually is a lot of the crop mixes that were talked about through research from some of the other previous speakers. And this is total acres over the entire 5 years. So not per year, but the total over the 5 years. So the canola pea is the biggest, so the old Piola mix. And then chickpea flax is the second at around 20,000 acres over the whole 5 years. Oats pea mix is kind of a close third. And then we also have barley pea and mustard pea. So you can see, there's the chickpea flax and then kind of brassica field pea mix and cereal field pea mix are kind of the common ones. And interestingly, these 5 kind of make up about half of the intercrop acres reported to us. The other half would be a whole variety of other mixes, of both two-way mixes and more complex mixtures, 3 and 4 crop mixes. So as far as insurance program development, so we are working on developing a more, tailored or specific insurance program for intercropping, but insuring any crop, including intercrop mixes, requires fairly detailed information on both the expected yields in order for us to set yield coverage, and also the variability in yields, and that's used to set the premium rates to have an actuarially sound insurance program. And so that information, like for any program we develop, is collected through a few sources. But so for intercropping, what we're reviewing the kind of small plot research results that all the things like all the previous speakers were talking about. So all University, Agriculture Canada, other ADF funded projects are reviewing all the research results on that, and also kind of monitoring intercrops grown kind of on a field scale by Saskatchewan farmers through kind of a new crops monitoring program that we have. And so we collect things like seeding rates, the resulting yields for the different crops in the mix, and also other agronomic factors or variables. And so if you're an producer that's growing intercrops and are interested in participating, you can get your crops monitored and help us by providing data. And if you'd like to do that, you can contact your local crop insurance office, and then that could be arranged. So it would basically be a monitoring 2 or 3 times during the growing season. And this is just a general reminder that the deadline is coming up March 31st, which is the deadline to make any kind of changes to crop insurance contract or apply for one if you don't have one already. And just general contact information. So that's if there's any questions now.
Wonderful. Thank you so much, Arlen. So we do have a couple of questions that are slowly coming in. So the first one is, what is your most common question about intercropping that you get from producers?
Probably when you could have a yield loss program, or can you develop a yield loss program? And the question or the answer, I suppose, is it's being worked on or developed, and like I said, collecting information to be able to set the yields and premium rates.
Awesome, thank you. So kind of jumping off of that point, so if you have an intercrop and one of the crops fails, but the other crop does really well, can you claim insurance on the poor crop?
For an intercrop, right?
Yes.
Well, it would be, the only way it could be insured is through our diversification program. So again, it's a proxy based on your other crops, so it's not based on the actual yield of the intercrop itself. So I guess the short answer is no. The longer answer would be, it would be dependent on your other kind of traditional kind of grains, oil seeds, pulses that would be insured, and kind of the average claim on those.
Awesome, thank you so much, Arlen. It was great having you present today. We really appreciate it.
Thanks for having me.
All right, everyone. So thank you all again to all of our speakers for this great information. We really appreciate the time that you guys took out of your day here today. At this time, we would like to thank everyone for coming to join us for the event. Don't forget to use the QR code that will pop up at the end of the presentation of the event if you are a CCA. If you have any issues or have missed out on the code but attended live, please contact us after the webinar and we will make sure that you get your credit. Credits are only available to those who watch the entire webinar live. Please take the time to fill out the survey that will pop up at the end when you exit from the program. This is the best way to get your feedback on what research or questions you have on intercropping so that we can continue to provide a spotlight to the topic in the future. Also, feel free to reach out to any speakers or local crop specialists with any questions. If anybody has any questions regarding these topics or anything else really related to farming in any capacity, please contact your local Sask Ag Regional Office or the Agriculture Knowledge Center at 1-866-457-2377, and we will be sure to put you in contact with a specialist to help you with your questions. Thank you all again for joining us today, and have a great day, and we'll be throwing up the QR code momentarily.